Hello students, welcome to Trunkie Academy. Okay, and today we'll study about the uh, biotechnology and about the introduction, how and different some basics idea about the biotechnological processes. Okay, then we go into details of each and every topic. Okay, and this is one of the important topic, and most of the students, they sometimes some students they try to avoid this top chapters. Okay, but this is one of the important chapter. Okay, so biotechnological process and principles and process, and apart from that there is another chapter called biotechnological applications. Okay, so both these chapters are important, and starting with this biotechnology principles and process, first we start with the introduction. Okay, what is biotechnology? The name itself you can understand that bio means life and technology means you know very well. So something the technology that related with the biology. Okay, so ever since the days of Rene Descartes, the French philosopher, mathematicians, and the biologists of 17th century, all human knowledge, especially natural sciences, were directed to develop technology. Okay, in 17th century. So that time the scientists, the biologists, and all the good educationists, they were in busy with some kind of discovery and the research, and they were all directed to develop technologies. Okay, and advancement of technologies. Okay, so which add to the comfort and the welfare of human lives. And nowadays you see technology is so advanced, and it has become so easier for us for different we have got washing machines, we have got the different kinds of gadgets that has become very much advantageous for us. Okay, and makes our works easy. Okay, so now Rene Descartes, the whole approach to understand the natural phenomena became anthropocentric. Okay, that means physics and chemistry gave rise to engineering, technologies and industries which are all worked for human comfort and welfare. So physics and chemistry that gave rise to engineering technologies. Okay, so that work for all human comfort and welfare. The major utility of biological world is as a source of food. Okay, so biology is a major utility. Okay, that the biology is a source of food, and also in medicine also. Biotechnology, the 12th century offshoot of modern biology. Okay, so biotechnology is actually the offshoot of the modern biology. So when modern biology developed, and biotechnology is a branch of science that came out from modern biology that changed our daily life, and as its products brought qualitative improvement in health and food productions. So when the biotechnology advanced, and when that came out of the modern biology as a branch, okay, so that changed our life, and because these advancement of the biotechnological advancement that brought about some qualitative improvement in health and also in food production. The basic principles underlying biotechnological process and some applications are highlighted and discussed in this unit. Okay, so according to our syllabus and the NCT guidelines, and also as per the NEET syllabus, we need to understand some important principles in the biotechnological processes. Okay, so Herbert Boyer, he was born in 1936 and grew up in a corner of western Pennsylvania, and he was a scientist, and he completed graduation at the University of Pittsburgh in 1963, and the postgraduate studies at Yale. Okay, so in 1966, Boyer took over the assistant professorship at the University of California at San Francisco. So this was about his education, and then in 1969, Boyer performed studies on a couple of restriction enzymes of the E. coli bacterium. So in 1969, he did some extensive research and studies on the couple of restriction enzyme is a kind of enzyme that breaks or fragments it's also called molecular scissors. They break the DNA into fragments. Okay, so the Escherichia coli is a kind of bacteria where we find which he used as the model organism, and from that bacteria E. coli he studied about the restriction enzyme. Okay, so Boyer observed that these enzymes have capability of cutting DNA strands. Okay, he observed and he found that these restriction enzyme, okay, these are able to cut the DNA strands in a particular fashion, which left was what has become known as the sticky ends. So after cutting the restriction enzyme and these restriction enzymes are also called the restriction endonuclease. Okay, the restriction endonuclease is a restriction enzyme. Okay, so after breaking the strands, what is left is the sticky ends. Okay, so that is ready to join with another one, the sticky ends. What will happen? EcoRI makes the sticky ends, so restriction enzyme, okay, or restriction endonuclease. When after cutting, it left the DNA with the sticky ends. You can see the sticky ends there, okay. So that is the sticky ends. This side. So this is the sticky ends. Okay. Now, okay, that it cuts after cutting. Now, see, these clipped ends or cut end made easy pasting together pieces of DNA. So this cut end is easy to pasting to the pieces of DNA. Now, Cohen studied, another scientist called Cohen studied small ringlets of DNA called plasmids in prokaryotic cell or bacteria. So he was a scientist who studied about the small the ringlet circular DNA called plasmid. Okay, so that was found in, that is the plasmid, that is the bacterial DNA, the plasmid, and he called this as the plasmid. Okay, so which float about freely in the cytoplasm of certain bacterial cells and replicate independently from coding strand of DNA. So that means they are independent of coding strand DNA and they float in the cytoplasm. Okay. Developed a method of removing plasmid from the cells and reinserting them in other cells. So this is a very important method and technique he developed. Taking out, removing the plasmid from the cell and then reinserting into another cell. Okay. So he discovered a technique of this removing and inserting the plasmid. Okay. Combining this process with that of DNA splicing enabled Boyer and Cohen to recombine segments of DNA in desired configuration. Okay, so and insert the DNA in bacterial cells. What they do after the technique developed by the Boyer, okay, and that means the cutting of the DNA into segments, into fragments by restriction endonuclease. Okay, so applying this knowledge, applying this technique, and also taking out, removing of the plasmids. Okay, so recombining these, okay, so Cohen these techniques. So Boyer and Cohen they recombine segments of DNA in desired configuration. So they recombine into desired desired configuration and insert the DNA into the bacterial cells. So what they did, took out the plasmid, okay, and they cut the DNA and then they joined into desired configuration and then they inserted into the bacterial cell. So that's what it had been shown in the picture. Cloning or expression vector plasmid, eukaryotic chromosomes you can see, and digestion with restriction endonuclease. So from after the portion from this eukaryotic chromosomes, that DNA fragments is joined and then ligation joining, okay, and then it is recombinant plasmid has been found and then it is inserted into the bacterial cells. So taking the DNA portion from the another cell or another chromosome, that is inserted into the DNA of the plasmid, and then it has been joined and inserted back into another bacterial cell. Okay, so which could then act as manufacturing plants for a specific protein. Okay, so that could act for manufacturing some kind of plant factory for the specific protein. Okay, so for example, if we wanted to, just for example, I am telling you the gene that is present responsible for synthesizing insulin protein. Okay, insulin is a peptide hormone. Okay, and if this insulin is not produced sufficiently in the body, so there is a rise of blood glucose sugar level in our body. So what happens when these the genes are taken, okay, and these are part of the DNA is inserted into the DNA of say and after making the fragments, okay, and we are making into a new recombinations we are creating, okay, taking out the plasmid from the bacteria and then inserting into that configuration, and that along with that gene which is responsible for producing the insulin into the bacterial cells. So that will start producing, manufacturing that particular protein which we have inserted the gene which is there for producing that protein. Okay, so that is the technique. This breakthrough was the basis upon which the discipline of biotechnology was founded. So this was the main foundation of the biotechnology, and here the main discipline of biotechnology was founded, and this started the it was a breakthrough, and from here so many different other discoveries and the invention and the researches took place, and that went to discovery of and the so many important things and made it easy for they played a very important role in the medicine and the improvement of crops. Okay, so biotechnology deals with the techniques of using live organisms or enzymes from organisms to produce products and processes useful to humans. In this sense, making curd, okay, bread or wine, which are all microbe-mediated processes, okay, could also be considered as a form of biotechnology, because we are using microorganisms, okay, or live organisms or enzymes from the live organism to produce the products that may be useful for the human beings or for humankind. Okay, so that is the process of biotechnology. So whatever like the making of the curd, bread or wine, okay, or cheese, this all comes may be considered as a result of a biotechnological process. Okay, so however the term is used in a restricted sense today, because this has advanced and it has advanced, and it has been so the many discoveries has taken place, and there's so many branches are there in biotechnology. So we do not nowadays the term is used in restricted sense to refer to such the process that which use genetically modified organism to produce products useful to human beings on large scale. Okay, like corn, soy, okay, cottonseed, papaya, rice, okay, rapeseed, and potatoes, tomatoes, dairy products, and peas. All these are different. These are the genetically modified foods. Genetically modified foods by tampering by changing or by modification of the gene, or you can say so by recombining the genes of these plants. Okay, and then getting the desirable product or desirable food. Okay, so this is the part. Furthermore, many other process techniques are also included on the biotechnology, and that is the in vitro fertilization leading to a test tube baby. So this is also comes biotechnology, because this involves the biotechnological process and techniques is called it also comes under the biotechnology. In vitro fertilization, with fertilization outside the body for the development of the baby, like test tube baby. When somebody is having infertility and there is no chance of fertilization in vivo, then in vitro fertilization is done for a test tube baby. Okay, so another example is synthesizing gene and using it. Okay, synthesizing a new gene and then using it to produce something. That also is the like in PCR amplification. Okay, in PCR is a polymerase chain reaction where the genes are made, so many different DNA fragments are multiplied. Okay, produce copies. So many copies are formed so that it will be easy to work on. Okay, developing a DNA vaccine. That is also very interesting and very important one. This is also comes under biotechnology. Okay, so apart from the genetically modified organisms, these are also the important the biotechnological process. So developing a DNA vaccine. So influenza virus is taken. So the plasmid is taken out. Okay, then influenza virus, the genes, plasmid plus genes, then insert. Then the vaccine is made, and this is the to protect from the disease. So correcting defective genes also comes under the biotech. So anything that is that includes the biology and the technology is the biotechnology. Very simply, if we can say, okay, because there is all in all this process we saw that these technologies, that some technologies are involved, and that is involved in the some biological process, or that comes under the biology. Okay, so it is all these are the biotechnological process. Now, the European Federation of Biotechnology (EFB) has given a definition of biotechnology that combines both the traditional view and the modern molecular emphasis. Okay, because we cannot ignore the traditional view also, and definitely there is a modern molecular emphasis. So, what is that definition? It is the biotechnology, the integration of natural science and engineering in order to achieve the application of organisms, cell parts thereof, and molecular analogs for products and services. So this is one of the best definition. Okay, that means the integration. Okay, combining joint integration of natural science with biology or other branch of science and engineering in order to achieve the application of organism, cell as parts thereof, and the molecular analogs for products and services. Okay, so now this means that biotechnology is a science which utilizes properties and uses of microorganisms or exploits the cells and the cells constituents at the industrial levels for generating useful products essential to life and human welfare. So that means biotechnology is continuously involved, and this can be involved in for the producing, or it is involved in for human producing the products that for human welfare, or for some kind of techniques to prevent from the disease, or providing some important medicines. Okay, so this can be considered as a branch of science that deals with or it utilizes the properties and uses of microorganisms or exploits cells and cell constituents at industrial level for generating useful products, and that is essential to life and the human welfare. Okay. Now we will go through some important questions. Who studied the small ringlets of DNA called plasmids? This was written by the Cohen. Okay, and not Spencer. It's not Boyer. It was the Cohen. So this is the important point. You should remember a ringlet. That is the plasmid. Okay. This was studied by the Cohen. In the year 1969, Boyle performed studies on, it was the restriction engine, restriction endonucleases. It's called nucleases or restriction enzymes. Okay, restriction endonucleases. So it's not endonucleases, it's a restriction enzymes. Okay, it's about it. Free floating ringlets of DNA in the cytoplasm of bacterial cells are called plasmids. Okay, it's not a plasmid, it's not cosmid, and it's all of this is the plasmids. Okay, and it is a circular DNA and it floats in the cytoplasm of the prokaryotic cells like bacteria. Now we come to the important principles of the biotechnology, the main, the principles that involved in the biotechnology. The two techniques that enabled the birth of modern biotechnology are that. Now the very important question comes here. The importance of the exam. Okay, there are two techniques that actually gave rise to modern biotechnology are the genetic engineering. Yes, the genetic engineering. Okay, understanding the gene, the genetic engineering, or we can say the engineering that involves the study of genetics and manipulating with the genes. Okay, for the human welfare. So one is the genetic engineering, and next is the tissue culture. So these two techniques were very, very important that gave rise to the modern biotechnological processes. Okay, or you can say modern biotechnology. So genetic engineering, okay, understanding the gene, okay, or gene configuration, or you can say the sequence and the DNA, and then manipulating or changing, or then the recombining of the gene, and another is the tissue culture. Okay, from where the small tissues of the cells, a group of cells, are cultured in the lab for production of the parts of the organism or whole organism. Okay, so tissue culture and genetic engineering are the two techniques that enabled the birth of the modern biotechnology. Okay, so now the genetic engineering, this includes techniques to alter the chemistry of genetic material, to change the so alteration, interfering with the genetic material, DNA and RNA, to introduce this material into host organism and thus change the phenotype of the host organism. So genetic engineering, what happens in they are changing the genetic constitution or genetic material. They alter the chemistry of the genetic material to introduce the material into host organism, okay, and thus change the phenotype of the host organism to change the phenotype, okay, that's the phenotypic feature. Now, nucleoid is there. You can see the PI plasmid, the agarobacterium tumefaciens is there. So, site at which restriction endonuclease enzyme cuts the type plasmid tDNA. Okay. Now, foreign DNA, the gene is inserted. Okay, gene of interest is inserted, and the restriction endonuclease DNA ligase. The ligase is then joined. Recombinant type plasmid has been formed and introduced into plant cells in the culture, so that to get the desirable phenotype, plant with new trait is obtained after this genetic engineering technique. Okay, this is how and this is an example how the genetic engineering, what is done in the genetic engineering. Okay, now tissue culture. This is I told you this involves the growth of only the desired microbe or eukaryotic cells in large quantities for manufacture or biotechnological products like antibiotics, vaccines, enzymes, etc. Okay, so that means when we are taking out some isolated cells, okay, and isolating the cells of our interest, and then we are culturing and growing in the lab. That is why the process is called the tissue culture. The sterile microbial contamination free. Okay, sterile microbial environment in chemical engineering process is essential for this. So there should not be any contamination. Then the result will be all not correct, and this will give a problem. Then will be the not desired result will be obtained. Okay. The conceptual development of the principles of genetic engineering. The sexual reproduction provides opportunity for variations and recombination genes. Okay, so that you have already studied in class 10. Okay, the variation is very, very important, and that is required for the evolution, and that forms the raw material for the evolution. Now, when sexual reproduction takes place, there is recombination of the genes. A new offspring has got some kind of variation as compared to the parents. Okay. So these variations take place from generation after generation, and ultimately their slow-going process called re-evolution is also occurring. So sexual development provides opportunities for variation and recombination of genes. Okay, some of which may be beneficial to organism as well as the population. But these variations is not always beneficial, or due to mutation, or the variation that comes with the mutation may not be suitable or maybe beneficial, okay, or maybe helpful for the organisms, okay. So in biological process we choose only the benefits. So our sexual reproduction preserves genetic information, while sexual reproduction permits variations. So what happens in the case of our sexual reproduction? The same set of gene comes, same set of the DNA comes to the offspring, and there is no variation. Okay. So the offspring and the progenies that formed after the asexual reproduction will have the same set, and there will be no change. So that means they have the same features and the same traits as their parents. Okay, and the genes that are present are also the same. Now, traditional hybridization procedures used in plant and animal breeding very often lead to inclusion and multiplication of undesirable genes along with the desired genes. So whatever in case of the hybridization procedures are used in plant and animals, okay, breeding, okay, often lead to inclusion or multiplication of undesirable genes also, because these genes are along with the desired genes. So while doing this hybridization process, okay, so that undesirable genes are also there, and that are going to show their phenotypes, okay, which we do not want to be expressed, okay. So what happens? For that we need to use our genetic engineering. So the technique of genetic which include is the creation of recombinant genes. So what we are doing? We are just cutting away the undesired DNA part segment or gene, and we are inserting, or we are using the gene which has we want to express the desirable product from that gene or part of the DNA segment. So what we are doing is they are taking the plasmid, okay, the gene for antibiotic, the green one you can see, the foreign DNA, region of the interest, okay, and this endonuclease or restriction enzyme plays the role for cutting. Now the sticky ends are there, and now we are after hybridizing process, the DNA ligase enzyme. Then we have to form the recombinant DNA. So that has formed the recombinant DNA after the choosing the particular gene we needed to form the recombinant. So we are taking that particular. So this is a technique of the genetic engineering. Okay. So plasmid is there, human DNA and the restriction enzyme cut, and then we got the insulin gene, as I told you earlier as an example. Okay, insulin gene. Insulin is a peptide hormone. Peptide hormone is secreted from the beta cells of islets of Langerhans of pancreas, and these genes are responsible for coding and expressing and for the formation of the insulin. So this insulin gene is taken out and is inserted into the plasmid for its production of the insulin. So we are doing this with the help of the DNA ligase. We are joining DNA ligase in the enzyme that helps to seal or join to the sticky ends, the cut part of the gene that we require to form a new recombinant DNA in the plasmid. Okay, so then this new recombinant DNA plasmid we are going to insert into the bacteria for the production of that the desired product, that is the insulin. Now, use of gene cloning and gene transfer. How we do that? Overcome this limitation and allow us to isolate and introduce only one or set of desirable genes without introducing undesirable genes into the target organism. So this use of genes cloning means what happens? Overcome this limitation and allow us to isolate and introduce and only one or set of desirable genes without introducing undesirable. So this is the advantage of this genetic engineering. Okay. Transfer of insulin gene. How? The restriction enzyme we are cutting. Okay, with the help of restriction enzyme cutting the gene. Then human cDNA is there and the linker DNA is there. Okay, so recombinant plasmid is formed after we are taking out the cloning of the. So why the cloning? Why do we need the cloning? Because we need more copies. So we are making the cloning, and it helps in the. So when they are replicating, they are forming a replica and they are getting the many transfer and cloning of the insulin gene. This is the process. Okay, so now. This is one of the technique of the genetic engineering. Okay. Now, a piece of DNA which is somehow transferred into an alien organism would not be able to multiply itself in the progeny cells of the organism. So, what we do? Bacterial cells, okay, and the bacterial chromosome is there. Okay. So, a piece of DNA which is somehow transferred into alien organism. Okay. So, what we are doing? We just but when it gets integrated into the genome of the recipient, it may be multiplied. So just by taking one segment of DNA or the gene of interest, for example, the gene that produces the responsible for the production of the insulin, if you taking out only that particular segment or gene and inserting into the alien organism or the bacteria, that will not be able to multiply. So what we need to do once we are integrating into the genome of the recipient, for example, we are making it integrated into the genome of that bacterium, okay, then it becomes functional and then it will multiply, okay, and into the host and that inherited along with the host DNA, okay. So what we are doing in bacterial cell and bacterial chromosome, the recipient, then we are integrating that into the DNA and then we are getting this is because the alien piece of DNA has become a part of chromosome which has the ability to replicate now, okay. So now, in a chromosome, there is a specific DNA sequence called origin of the replication, which is responsible for initiating replication. So, what happens? There is some particular region in the DNA. Okay. So there is one region called origin of the replication, which is responsible for initiating the replication. So, origin of replication, there is a site or there is a region. Okay. So replication forms newly synthesized. So replication sport or replication initiates or starts from the origin of the replication site. Okay, and then there is a formation of replication sport. Then newly synthesized DNA will start forming, and then there is a replication text, replication of the DNA text, whole replication of the DNA text. Therefore, multiplication of the alien piece of DNA in an organism is not possible unless it becomes a part of chromosome which has a specific sequence known as origin of replication. That is the main reason, okay? It's not because the DNA, there is a one region called origin of replication. So when it becomes a part of the host organism, okay, or host chromosome, so there is a part of the region called origin of replication that becomes initiated. From there, it can be replication can be initiated. So that is the reason we need to integrate. Thus, an alien DNA is linked with the origin of replication so that this alien piece of DNA can replicate and multiply itself in the host organism. So this is the reason an alien DNA is linked with the origin of replication. So it is, yeah, when while integrating, we are integrating with the origin of replication, so that replication processes start and multiplication of the host cell takes place. Okay, in that. Now this process is called cloning, or making multiple copies or multiple identical copies of any template DNA. Cloning, just copying, like in Xerox or photocopying. Okay, so making a template DNA. So copying so many, the multiple copies. This is a cloning. Okay. Now, construction of the first artificial recombinant DNA molecule was there. The first recombinant DNA was considered by linking a gene encoding antibiotic resistance with a native plasmid autosomally or autonomously replicating circular extrachromosomal DNA of Salmonella typhimurium. Now, this was the first artificial recombinant DNA molecule was made for what? By linking a gene encoding antibiotic resistance, okay, antibiotic resistance with a native plasmid autonomously replicating circular extrachromosomal DNA of Salmonella typhimurium bacteria. Okay, now try to understand this one. Okay, there is a bacterial plasmid is there, and the foreign DNA is there. Okay, then the restriction endonuclease has cut. Okay, and then we have got the same concept. Okay, the cloning. Okay, because we need to make many copies. Okay, after we have integrated it. Okay, now Stanley Cohen and Herbert Boyer accomplished this in year 1972 by isolating antibiotic resistant gene by cutting out a piece of DNA from a plasmid which was responsible for conferring antibiotic resistance. So while studying in the bacteria, they were able to isolate. Okay, and they isolate and cutting out a piece of DNA which from a plasmid which is responsible for antibiotic resistance. Okay, so this is how they did. So this was an important advancement in the biotechnological discovery. Okay, and this was an important advancement in the biotechnology. The cutting of DNA at a specific isolation became possible with the discovery of the so-called molecular seizures, that is the restriction enzymes. So restriction enzymes are actually the restriction endonuclease, which are able to cut the DNA into fragments at specific sites. So that is the definition of the restriction enzymes. Now, restriction enzyme cuts DNA into gene site pieces, small pieces, and a single gene of interest is considered. All of this happens in a test tube. Okay. So restriction enzyme has cut and we got the fragments. Okay. The cut piece of DNA was then linked with the plasmid DNA. Okay. Then the cut DNA. Okay. The cut part of the DNA that is linked with the plasmid DNA. Okay. The restriction enzyme cut. The gene of interest is taken. Then it has been integrated with the plasmid DNA and the gene of antibiotic resistance that was and with the help of the DNA ligase. Okay. Now the modified plasmid has been formed. Modified plasmid has been formed after this process. Okay. Now these plasmid DNA acts as vectors to transfer the piece of DNA attached to it. So these plasmid DNA is actually considered as or regarded as the vectors, okay? Because they are involved in the transfer of the DNA of interest, okay? So we are considered this as a vector, okay? Now recombinant DNA, a piece of DNA, okay? And now bacterium, which is the bacterium in binary fission is taken place, and now there is two copies. We have got two copies. Okay, thus we are getting. So this is how the vector is involved in the formation or making the copies of the desired DNA. Okay, so this plasmid, the first bacterium in which it is inserted, this is the vector. Okay. A plasmid can be used as vector to deliver an alien piece of DNA into the host organism. So this plasmid can be used as a vector to deliver an alien piece of DNA into the host organism. So this linking of antibiotic resistance gene with plasmid vector became possible with the enzyme. Now another point is very important. Now if there was a discovery of the restriction endonuclease or restriction enzyme, okay, with the help of which we were able to cut the DNA into fragments. But what about the joining? If it is cut, we have to join also with the gene or the fragments or the part of the DNA of our interest should be we need to integrate and join with the plasmid. So how to do this without the DNA ligase? So important step was that in the discovery, the DNA ligases also play a very important role in joining that part. Okay, so inserting that the DNA ligase. Now, in this case, this enzyme acts on the cut DNA molecules and joins their ends. So they helps in joining the fragments of the DNA. Okay. This makes a new combination of circular autonomously replicating DNA created in vitro, okay, in the lab, okay, and it's known as recombinant DNA. So we are taking the DNA of our interest and then we are integrating with the help of the DNA ligase into the plasmid DNA, and then we are getting a new DNA plasmid, that's a recombinant DNA, okay, like cloning or expression vector plasmid, digestion with the restriction endonuclease. Then we are all from the eukaryotic chromosome, we are getting taking out the DNA fragments with the of our interest and with the help of the restriction endonuclease, and then again we are placing this into for the ligase in the plasmid, okay, or the bacterial DNA. So this forms a new recombinant plasmid. Now when this DNA is transferred into E. coli or Escherichia coli, a bacterium closely related to Salmonella, it could replicate using new host DNA polymerase enzyme and make multiple copies. So what happens when this DNA is transferred into the Escherichia coli, a bacterium closely related to Salmonella? Okay, so this Escherichia coli and Salmonella are little closely related, and it could replicate using new hosts. Okay, and host DNA polymerase enzyme and make multiple copies. So transfer. So this was into Escherichia coli. Okay, from there. So multiple copies with the DNA polymerase enzyme. Okay, using that host DNA. Now bacterial DNA, plasmid, and the cell replication. The ability to multiply copies of antibiotic resistance gene in E. coli and was called cloning of antibiotic resistant gene in E. coli. Okay, so you can see the same diagram. Okay, now cloning. Okay, so this ability to multiply copies of antibiotic resistant gene in E. coli was called cloning antibiotic. Okay, cloning of antibiotic resistant, because we are producing multiple copies of antibiotic resistant gene. We need antibiotic resistant genes. So what we are doing is we are making the copies of that by cloning. Okay, so this is the cloning of the antibiotic resistant genes. Now there are three basic steps in genetically modifying an organism. Identification. First, we need to identify which gene we require, which gene is desirable, and which gene we need to which is the gene that is of our interest, as we need to do some we need to undergo the biological techniques. Okay, so first identifying the DNA with desirable genes. Second is the introduction of the identified DNA into the host, and the third is the maintenance of introduced DNA into the host and transfer of DNA into its progeny. So these are the steps. Okay, and now we go through quickly through the questions. The techniques that enabled the birth of modern biotechnology are the genetic engineering and enzyme technology, genetics and the rDNA technology, tissue culture and disease resistance, or genetic engineering and tissue. Number 4, genetic engineering and tissue culture. These are the two important processes that led to the evolution, or you can say the advancement, or you can listen of the modern biotechnology. Okay. Now, number 2, the specific DNA sequence in the chromosome which is responsible for initiating replication is that is the promoter origin, or origin of replication, or Tata box region, or it is the downstream region. It is the origin of replication where the replication starts, so the replication begins. Okay. The third is that the molecular seizures in genetic engineering is the proteases, or it is the lipases, or it is the esterases, or it is the restriction enzyme. It is the restriction enzyme that is the restriction endonucleases. Okay, so it is the restriction enzyme. Now, the function of DNA ligases is to cut the DNA molecules, or is it to link the DNA molecules, or it is cut and link of the DNA molecules, or all of them? No, it is ligase is to join the DNA fragment. So the answer will be link the DNA molecules. So number 2 for this question will be the correct option. Okay. Now thank you. This was all about the introductory idea and the important terminologies and about the biotechnological process. What is biotechnological process? What are the different the basic principles involved in the biotechnology? Okay. So this was all about the important basic points about the biotechnological principles and the processes. Tomorrow again we will meet at the same time to study in detail about the different biological techniques and the processes. Okay. So after this class, I will send you the DPP like I have already told you. Again, I am repeating the same thing that when you go to your academic booster, when you click on your resources, you will get another link. There's so many other links, and you'll find there is a link called writing material. When you click on that, you'll find the DPP. DPP means the daily practice problems or daily practice problems. Then click on that the DPP or the topic taught today, and then you'll get some questions and go through the questions and answer those questions and send back to us for evaluations. Okay, so thank you for joining today and listening, and tomorrow again we'll meet at the same time. So till then, goodbye and thank you.